pointer. Okay, here we are. All right, so let's go back over some of the same material. Now, why so much redundancy? Well, some of you are jet lagged. Redundancy is not a bad thing in general. But also, as I go over these topics, many of which have been introduced by Joe and Pat, I'm going to show you landy-land notations for everything. So we're going to introduce the G matrix and things like that as we go into this lecture and into two characters. Okay, and so this little icon here is to remind us, as Joe was pointing out, that a lot of the developments that we now use in evolutionary quantitative genetics come from this application in plant and animal breeding, where response to selection and improvement of various phenotypic aspects of plants and animals was the goal. And we're going to adapt that. Gee, can I even do this? No, that's because I haven't plugged this in. Okay. All right, so here's our thesis, and we're just going to you can go into the notes and drill down on these topics in great detail. I'm just going to keep it at the very most basic level so we have simple takeaway. First of all, point one, here's our thesis. Most traits are affected by many genes. This is the point that Patrick was making, among others, in his talk. Secondly, we can model the inheritance of such traits with a statistical approach. Even if we've got thousands of genes, we can still use the statistical approach derived from Fisher's insights. Thirdly, in that Fisherian approach, additive genetic variance is the statistical concept that enables us to model both inheritance and response to selection. It also enables us to model drift in the trait as drift changes the mean from generation to generation. For all of these things, inheritance, that is to say resemblance between parents and offspring, response to selection, response to drift, genetic variance is the key concept. And so that's why we're so much more interested in additive genetic variance than we are in dominance variance. There are relatively few things that dominance variance is good for, but additive genetic variance is useful in many different ways. Okay, so here's what we're going to do. We're going to go back again and look at phenotypic resemblance between parents and offspring, and we're going to look at, again, the Fisherian point that it reveals heritable variation, enables us to estimate additive genetic variance. And then we're going to go back and look at our model. We're going to look at a slightly more elaborated model in which we include not only dominance but epistasis. And then we're going to come back to this question that was raised by one of you, is why don't we run out of additive genetic variance? And then we're going to finally look at how the mean is changed by selection from generation to generation. So a review of some concepts we just talked about and a little more detail on some of them. Okay, here's Galton's plot. We've flipped the axes so that we're looking at offspring height as a function of parental height. And Galton wants to take all of these data on stature in the British population, and he wants to crunch it onto one graph. That's what he was trying to do. So you've got two parents, so he takes the average of the two parents. So here are parents that look like their average height was about 69 inches. That would be 5 foot 9 inches tall. I'm sorry, we're in British units. And this is the average height of offspring. And I can't remember what we do about, I guess we're probably averaging sons and daughters, I think is what we're doing. Okay, so we're still using this graph today, and here's the line of perfect inheritance in the dot line, and here's the actual regression line we put through the data. And what Galton is pointing out is that if the parents are tall, then the average offspring tends to regress upwards towards the mean. If the parents are exceptionally small, then the average offspring tends to regress down towards the mean. All right, I'm going a little fast here, but all right. So there's Galton's plot. And what Fisher was trying to do, and what we're trying to do with this model, is explain that plot. we see an overall, and it looks fairly weak, but the regression line's quite strong in that plot. If you notice, it's something like about, what is it, about 0.6, something like that, 0.7. I forgot to, let's go back. Do I have it on the graph? Yeah, okay, so look, here's the regression slope, right? And what we saw, what we're going to come out of this with is this slope, mid-offspring on mid-parents, is going to estimate the heritability of the trait. And it's about 0.6. Now, we need a model for what's going on here. Because when Galton was doing this plot, he said, look, there's resemblance between parents and offspring, and here's a way of summarizing it. We'll just summarize it with that regression slope. And the problem that Fisher, Wright, and Weinberg tackled was, where does that regression come from? What is that regression? What is that resemblance between parents and offspring an expression of? And so Fisher essentially went out and built an underlying Mendelian model to explain that covariance. And he starts by saying, let's look at this. This is Joe's model. Let's take the phenotypic value. Here we're calling it P, and we're going to say we've got contributions from loci A, B, C, and D. And if we know genotypes, we take this value for this contribution from locus 1, say this contribution from locus 2, this from 3, depending on what the multi-locus genotype is, and we have an environmental contribution, which we draw is a draw from a normal distribution. We add all those things up. Sorry, this is the locus E, and then we add an environmental effect, and we can get. This is our model for phenotypic value. Okay, now we're going to change notation, and let's call phenotypic value, and henceforth in my lectures, I'm going to always call it Z, following Landy's notation, Landy's paper. And we're going to think of phenotypic value as composed of just two things, an additive genetic value and an environmental contribution. We're going to call the additive genetic value X, and it's going to be the sum. Remember, Joe did this in one of his steps. So let's take, at each locus, let's take the additive effect of that locus, and we're going to add those all up, and that sum of those additive contributions we're going to call X. And that is also known as the breeding value for this particular trait, the sum of additive effects. As Joe pointed out, we're going to assume that these breeding values and the environmental values are independent. And so when we look at means and we look at the second moments, things are nice and additive. There are no covariance in terms out here because we've assumed that they're not there. And so in particular, if we look at the variance in phenotypic values, we're going to call that P, right? Notice how inconvenient that is up here, right? But we're going to call that the phenotypic variance, as if we were taking that, we could estimate it by taking the variance in phenotypic values from individuals in our population. We're going to have the variance in breeding values. We're going to call that G. We called it VA before, and historically that was true. And the variance in environmental effects we're going to call big E. And by the assumption of independence up here, the variance is the sum of these two independent genetic and environmental parts. Okay, so that's going to be our summary model and an introduction to the notation we'll use. Could be a better way of doing this. Okay, and as Joe pointed out, that gives us a new perspective on Galton's regression. Because if we look at the covariance between an offspring and one parent, and we work, well, let this be let this be mid-offspring and let this be mid-parents. We work through the algebra, we end up saying that this covariance between, this phenotypic covariance is equivalent to the covariance between the breeding values of offspring and parents. There's no covariance between E's because we've assumed there's no environmental covariance between generations. And so that covariance is equivalent to the genetic variance in the trait, right? This covariance is equivalent to the variance in breeding values. And we're right back where we were. This is heritability. I'm not going to write heritability because in the lectures that follow, when we go to the multivariate case, this is going to be the equation we're going to use. Well, sorry, this is equivalent to heritability, but this univariate expression doesn't generalize to multiple characters, but this does. Well, I'm getting ahead of myself. At any rate. Okay, so what comes out of this model then is an identification of genetic variance as the key concept that's explaining when we see resemblance between relatives, between parents and offspring, we're looking at the manifestation of underlying genetic variance under some assumptions that there aren't other causes that there aren't non-genetic causes of resemblance. All right. So someone asked about epistasis. In the formulation that we looked at before, we were just ignoring dominance deviations and epistatic contributions to phenotypic value. And basically the reason we're doing that is we're going to be primarily interested in the covariance between parents and offspring to look at responses to selection from one generation to the next. And so what we're going to look at here is the fact that when we look at, even when we include epistasis, the phenotypic covariance between offspring and parents is largely due to genetic variance and not to dominance variance and not to an epistatic contribution. So if we have a model like this in which we've already talked about dominance deviations, right? This then is the sum of those dominance deviations that Joe talked about across loci, just like X is the sum of the additive effects across all loci. Similarly, I is going to represent the contribution of epistatic interactions between pairs of loci. And this is Cockerham's formulation in 1954. And what Cockerham is pointing out is that if you break out all of these possible epistatic contributions from the loci, you can have contributions that come from the interaction between additive effects at pairs of loci, and we're adding those up across all pairs of loci. Then there might be epistatic interactions that you can think of as interactions between additive and dominant effects at different loci, and we're adding all those up. And then here, let's go clear out here. Let's imagine that we've got triplets of genes, and we're looking at additive by additive by additive interactions, right? So anyway, you can decompose the epistatic contribution to an individual's phenotype is coming from all of these different terms. And then when you break out the covariance between a pair of relatives, X and Y, it's due to all of these terms. And in particular, what we're calling R here is what Joe called F1 in his lecture. And then there's a coefficient U, which is equivalent to F2. This is the probability that alleles in two individuals are identical by descent. And this is the probability U, F2, that a pair of alleles in two individuals are identical by descent. Okay, so the point is, we're primarily interested in the covariance between offspring and parents because that's the key through additive genetic variance, to looking at resemblance between parents and offspring that's going to figure in our response to selection formulation. We can see that because U between parents and offspring is 0, that this term drops out. So does that one, so does that one, so does that one, so does this one, and on out in our series, right? All of those terms that involve interaction, that involve dominance deviation or epistatic interactions between additive and dominance parts, all of those drop out, right? So if we look at the covariance between parents and offspring, it's just due to this term, additive genetic variance, and this term, the genetic variance due to additive by additive epistatic interactions. But notice we're raising a fraction to a power here, right? So in the case of parents and offspring, we're squaring it, so that's 1/4. So this is likely to be a relatively small term compared to this one. And so often in the work we know by algebra we can forget about dominance contributions and looking at the covariance between parents and offspring, and probably the contributions of epistasis are relatively small. Now, Adam, I think, can imagine examples in which this might be large, but we'll talk about that. All right, so that's our point. The justification for the concentration for much of the rest of the course on additive genetic variance, and for right now, we're going to ignore epistasis as well as the contributions of dominance. All right, so here this topic came up. What about using the theory in something besides the barnyard and the greenhouse? Here's an example from my own work. In the garter snakes that I've worked on, you can count the number of vertebrae in the body, and you can, as distinct from the number of vertebrae in the tail. So if you look right here, if you're close to the screen like me, you can see the ribs end right there. So this is the beginning of the tail, so we can count number of tail vertebrae and number of body vertebrae. And you don't have to do an X-ray to do it. If you flip a snake over, you can see the ventral scales, which line up 1 for 1 with the body vertebrae, and there's a distinct set of scales on the tail that are divided, and they can give you the vertebral count. Now, the lovely thing about this is, when you hatch from an egg or are born, as in the garter snake case, you've got your adult complement of vertebrae right there. You don't have to raise them to maturity. At any rate, if we look at the distribution of body vertebrae, we see something very close to a normal distribution. This is not a huge sample. It's something like, I don't know, 150 individuals, something like that. And then if we look at a resemblance plot, we get something like this. If we plot, now there is sexual dimorphism, so we're going to treat the sexes separately. So here's the daughter's body count against the mom's body count. This is a single parent here, and then this is the average count of all the daughters. Typical litter size is about 10, so on the average there'd be 5 daughters that would go into each of, that would represent 5 daughters and 1 mom going this way. Okay, so we can do the same thing for the tail. And in both cases, we see a positive regression line. And if we just estimate if we estimate heritability as twice this slope, it's 0.54 for the body vertebrae, and about the same, a little less than 0.5 for the tail count. Let's see if I've got my animation coming up here. All right, so what we're going to do in the afternoon is we're going to look at a bootstrap first of all, we're going to estimate these regressions for these counts and some others that I'm going to show you using the mother-daughter data set, show you how to use a program to estimate these regressions. But we're also going to show you some R script that will enable you to bootstrap the sample and do a bootstrap estimation of the slope. And we'll do a sort of crude animation so you can see how that line flips around under bootstrap sampling. It sort of gives you a visual impression of what confidence you might put in that regression slope. And you'll see the confidence is pretty good because basically the lines are going to go like that. Or you could calculate standard errors or something like that if you wanted. All right, so there's a real life example. Now if we go into the literature and just pull in heritability counts from studies that have been done for morphological features. This is a summary that was produced by Derek Roth, and it's based on a total of 580 samples. For morphological characters, you can see a spread that has sort of a mode about 0.4. In other words, the average morphological study, these are usually measurements or counts, commonly yields a heritability of about 0.4. And years ago when I was doing a sabbatical leave in Scotland and talking to Bill Hill, he said, I was talking about comparing heritabilities for my my vertebral counts in different populations. He says, it's always going to be about this. It's probably going to be about the same. And he said plant and animal breeders like cow breeders that he worked with, for a conformational trait, size or something like that, typically the heritability is about 0.5. And that's borne out by these studies. Now, you notice you even get some estimates that are greater than 1, right? And even less than, and even negative, right? But those are sampling errors. The parameter can only vary between 1 and 0. But in any case, when we go to, you're going to look at the phylogeny of your lizards and maybe want to model the evolution of body size, and you're going to need a heritability estimate to use. You have no heritability studies for your genus of lizards. Suppose no other close nearby genus has an estimate. A reasonable thing to do would be to use a modal value for heritability of 0.4 on the basis of that survey. Just as a way of connecting. All right. Somebody asked this key question. This is a good question. Why don't we run out of additive genetic variance? And as a matter of fact, we don't. What happened to Patrick Phillips? He took a break. Oh, you're back there. So we're not running out of additive genetic variance in most selection experiments and in most laboratory, barnyard, and greenhouse. And there could be multiple reasons for that. But one reason is that we need to think about the ongoing, never-ending supply of new variation coming in from mutation. So if you think about mutation, it's not obvious that it's a substantial input each generation. But if you stop and think that each trait might be affected by dozens, hundreds, perhaps thousands of loci, then that what's a relatively rare event on a per locus basis can be a substantial source of input each generation. And then we've got loss of genetic variation each generation from stabilizing selection. So why, and from directional selection, but why would we put up stabilizing selection in this diagram? Why would we focus on stabilizing selection? I do know. But what do you mean by that?
Because normally you have a mean phenotype that has more advantage, and if you go too much outside that, you die, basically.
So another way of saying that is that's true, but that mode of selection we think is relatively common in natural populations. Now, when we get into the selection studies, we're going to show that actually disruptive selection is almost as common as stabilizing selection. But a stronger justification for thinking about stabilizing selection as maybe the primary mode is the observation in the fossil record and even on shorter time scales that the means are very stable. Stasis in most traits is the common evolutionary mode, and deviations from stasis are the exception. And the easiest way to account for stasis is stabilizing selection. So when we want to think about what's maintaining variance within genetic variation within populations, a balance between mutation and stabilizing selection is a very reasonable model. Now, it turns out when you make a model of that, you end up being a little bit short on genetic variation. I'll show you how we might deal with that in the next slide. Stabilizing selection is also going to create linkage disequilibrium between because you tend to link alleles that have an effect that pushes you towards the center of the mode under the biggest fitness advantage is being close to the mean. So there's selection that creates these link combinations of alleles. And that, by recombination, is going to be broken down. So you're constantly shifting between these two expressed and hidden sources of variation. So what about this model having a shortfall? I personally do not worry about that too much because in the real world, our populations aren't closed systems. And in particular, we know that there's a lot of migration coming into the population and even hybridization. So mutation isn't the only ultimate source or isn't the only source of new variants coming into the population. And also in finite populations, we're losing things by drift. So when we think about what maintains genetic variation in the population, the long-term equilibrium is probably a very complex process involving all those kinds of factors. So the idea is, and this is due to Landy in one of the papers listed at the end of the talk, is that we might equilibrate our expressed variation in some kind of a model like this. And indeed, if you look at the fossil record again, it's often not only the case that the mean is relatively stable, but also the variance, the phenotypic variance, is relatively stable too. All right, we're screaming along. Okay, let's go back and look at changing the mean with selection. Again, we're going right over the same territory that Joe did. I'm going to do it with mid-parent values and mid-offspring values. And so in this case, when we calculate the slope of the line, it's estimating heritability, not 1/2 the heritability as it is when you regress offspring on one parent. So let's do the thought experiment in which we say, let's suppose there's truncation selection so that only the parents over this threshold become the actual parents of the next generation. And then what's a key index for us is the difference between all potential parents, that is the blue plus the black, and black. So if you look at that difference between the trait mean for all parents and the trait mean of the actual parents is what we're going to call the selection differential. And so just as this regression line is the regression of parent values on, the regression of offspring values on parent values, if we take a change in parent values, we can use the regression line to predict the change in the expected change in offspring values, right? And lo and behold, that gives us this expression for how much we expect to change the mean in a single generation by selection. And so this is what we're referring to as the univariate breeder's equation. Now, why is this going to be useful in natural populations, right? This seems to be a model for truncation selection. But as a matter of fact, we can have a sample of parents that are the parents that survive, for example, and the sample we're looking at doesn't have to be just formed by truncational selection on some sort of a threshold, but we can have any weighting function we wish generate the parents that are going to be the actual parents. And the difference between all parents and those parents is still going to be the selection differential. So this breeding equation doesn't just work for truncational selection, but for any selection mode you want in which you can calculate the trait mean of the parents after selection. So that produces the generality that becomes useful to us. Okay, so here's an interesting point. If you think about our little equation that we wrote, this produces what Gould called in one of his books the boring march of the frequency distribution, right? So you got a phenotypic distribution, you go selection, then we do, same selection differential next generation, and the frequency distribution marches across the page generation by generation. And it is kind of boring. And the trend line for the mean is shown in this white line. But what if the population is finite? If the population is finite, that means there's, just by accidents of parental sampling, even though you're selected, the selection differential is actually fluctuating under this parental sampling scheme, right? And as a consequence, each generation there's a contribution of drift of the errors in parental sampling in addition to the expectation you'd expect just in a deterministic model. And so through time, you've got some lines that are doing, look, here's an extreme one and here's an extreme one down here. The lines are distributed about the trend line. And it turns out they're normally distributed. And that seems strange at first, but you've got all of these little contributions, right, that are adding up over time. And by the central limits there, we're expecting the distributions to form a normal distribution at any time slice. And they're going to be expanding like that. It's amusing to look at the animation, I think. I think I say yes. But we go over there. What do I want to do? You're not going to do my link for me? Do I really have to minimize this to see it? Jeez. Where's my screen? So cruel. All right. And I'm just trying to recover my, here we go, available files. Let's go back there. All right. You can get it to work on your, I don't know why it's not, it works in the PDF and it works in the PowerPoint, but not in this particular, anyway, I'll work on that tonight. All right, let's see, where do I want to be? I want to be, I don't know why I didn't have time to practice on this configuration. Okay, let's go up a few slides here. Anyway, there's an amusing animation for you to watch. All right, and indeed, when you do selection experiments, for example, here we're selecting on abdominal and sternal and sort of thoracic bristle counts in Drosophila, and you can see that they're not just straight lines, right? Your experimental populations are finite, and so the lines are drifting around. And it's the fact that these are all the down lines and these are all the up lines. It's the fact of all the bouncing around that tells you that there's a drift contribution. But the fact that you've got a difference between the up and the down lines tells you that the direction of selection certainly is making an effect. A difference, and our best estimate of the selection response would be the middle of that distribution of line means. Now you'll notice that even though selection is constant, in these data, it looks like we reach a selection plateau, not so obvious up here. But in fact, going up, we seem to have less response than going down, even though the selection differentials imposed each generation are the same. And so effectively, something is resisting the selection differential, maybe natural selection. Maybe it's really bad to have more than 20 bristles. At any rate, there are multiple explanations for it. So this is an example of just using just the Breeder's equation. We should have symmetrical responses, and clearly we don't. So even in the theory is even in a laboratory setting is good for a qualitative prediction of what's going to happen, but it's common to get deviations that are telling you something is left out of the model. Countervailing selection that's opposing your deliberate selection, or the possibility that there's just a difference in expressed variation at this end of the scale compared to this end of the scale, expressed genetic variance. All right. Okay, so we've tried to keep to the general points here in doing this fast, and here are the main things we've learned. We've made the point and illustrated that G is the genetic variance, added to genetic variance, is the key to understanding resemblance between parents and offspring. There might be a jigger of additive by additive epistasis in there, but maybe that's not that important. And because of that, G is also the key to modeling responses to selection and responses to drift. And our third point is that, and I love this verb. This is Jay Lush, who I think from a long career of teaching had a very nice evocative choice of verbs. As Jay Lush pointed out, genetic variation is nibbled away each generation by selection, but it's restored by other processes, in particular by mutation, migration, perhaps by hybridization, depending on the natural setting. And I like to do this too. I picked this up from Joe. Here are sort of key references to this lecture, and some of them are available. I think I put some of them up as PDFs as well.
Thank you. I wonder if you can comment about the comparisons of heritabilities of different, for instance, traits.
Asymmetry in the response?
Yeah, related to
there?
the review of Musso and Roth 87. What's your comment about this pattern of different kind of heritabilities, life histories, lower,
in the summary slide, you mean?
Right.
So it's been a while since I've looked at it. Do they get the sort of expected result that you get less heritability for life history traits? Is that what they got? I can't remember.
Yeah, that's it. And they classified different kind of traits of large sample of publications.
Yeah.
So they behavior has less heritability than physiology, and physiology has less than morphology, and this made me wonder about the specifics of variance component from a given population that Joe commented before. So what's your impression about that?
Well, so I think intuitively you can imagine, life history traits being components of fitness are probably perpetually under directional selection. And a lot of traits like vertebral numbers and probably body size are probably under stabilizing selection and maybe close to their optimum. So you can imagine that the life history traits are under stronger selection and more genetic variation is getting nibbled away, and you might expect less genetic variance and perhaps less heritability for that reason. But there's there are also differences in mutation and that could come into play. And so I think it's actually a little hard to make those, if your prediction is fulfilled or violated, I don't get too upset about it, and I'm not too thrilled if we show that morphological traits have higher heritability. I mean, the prediction's really about the genetic variance in the first place, right? Rather than this ratio. So you'll see in one of the things that I suggested that you read is that as soon as we go to multiple traits, heritability ceases to be a useful concept. It's because now you can go into it tomorrow or later in the afternoon when we talk about that, and so. That makes me think that even in the univariate case, it does turn out that when you write the Breeder's equation, you can write it as having a denominator of phenotypic variance. But there's another way to look at that phenotypic variance as well, the role that it plays. So I think you want to talk about the equilibration of and the evolution of genetic variance, or the G matrix, rather than that ratio. But so this is our third point, is that you want to think about why do we have genetic variation, what accounts for its stability or lack of stability. It comes down to a balance between opposing forces and whether we're equilibrated or whether we're thrown out of that equilibration by the events of the moment. Or just the finite size of the population. Yes. Oh, do you have we'll get you a...
Is stabilizing selection different from purifying selection?
Say again?
What's the difference between stabilizing selection and purifying selection?
Stabilizing selection and what?
Purifying selection or negative
Oh,
selection?
Purifying. Typically, people dealing with quantitative traits say stabilizing selection. In molecular evolutionary genetics, people refer to the elimination of extreme variance as purifying selection, but it's basically the same thing. I think Joe used the term optimizing selection instead of stabilizing selection. So, and there are other terms as well. Simpson, for example, refers to centripetal and centrifugal selection. If you're a little bit dyslexic like me, it's like that's just too hard to keep track of.
Thank you. Would you consider things like fluctuating selection under, I guess, stabilizing selection broadly?
Yeah, sure. So what we're thinking about, so what we're going to do tomorrow, I think, is we're going to think of selection in terms of selection fitness functions or surfaces or curves, and then the question is, what do they do from generation to generation? How do those functions change? And they're certainly going to fluctuate, all of them, but and there's of course an empirical literature on that. And of course we can build that into our simulations and models too. Okay, yeah? Oh yeah, you do. All right, oh look, we got two up here. I'd like to toss this to you, but our high performance guy wouldn't like that. There's a little button and it'll.
So the question I have, going back to your response to selection slide, the one before this one.
You want to go to back? Oh, you mean the
bristle number.
bristle numbers? Yeah.
Yeah. So
It's always nice to look at the real world, isn't it?
yeah, so it looks like, depending on your force of selection, the evolvability is going to be asymmetric. Is that a reasonable
Well, you know what? So
conclusion?
what's happened? These are very simple-minded experiments in the sense that you're doing truncation selection, and every generation you're trying to impose the same intensity of direction. You know, the truncation point just moves over as the frequency distribution marches. If you're with me.
But the reduced response that you see
Yeah.
in certain directions, is that going to be reflected in different evolvabilities, do you think?
Well, you're certainly getting, in the sense that you're not getting the same response to selection. I guess you could say evolvability is greater in this direction than in that direction,
Right.
if you wanted to say that. Now, why that's true
Right.
is ambiguous just from this data.
Mm-hmm.
So, in other experiments, what people will do, they'll get something that looks like the approach to a plateau, and they'll say, what if we take this line and subdivide it and then select down? You know, do we get a downward slope like this, or do we get a downward slope like that? And the general answer, typically it's like this. So there's something about that direction of selection in that part of that direction of selection rather than the trait at that position. In other words, it can move rapidly down if we, you know, yeah.
That makes sense.
So that's informative.
Thank you. Yeah.
So one practical reason that you frequently see asymmetrical responses to selection is that in one direction, in addition to selecting on whatever trait that you're interested in, you also end up selecting for decreased longevity in some fashion. Right, there's a correlated response in a life history trait that you can't get beyond. And so, for example, I worked on a large selection experiment in mice selecting for high voluntary wheel running activity. And we decided before we even began, from preliminary data, that we weren't going to select down because we knew we were going to be selecting for poor phenotypes and that it was going to be hard to maintain those lines. So we ended up increasing the number of replicate lines for the up selection and for the control, but we completely decided not to do the down selection. So that's a practical aspect of a lot of selection experiments.
So the summary is that asymmetrical responses are pretty common, and I think the consensus is there's a grab bag of explanations for what's going on.